When we talk about treating anxiety, OCD, or insomnia, the conversation almost always starts with a chemical imbalance. The standard model gives us a simple picture: serotonin or GABA floating between brain cells, passing calming messages across the synaptic gap. But that model leaves out a massive physical barrier. Those neurotransmitters do not actually cross into the target cell. They hit the outer membrane and stop. The signal still has to be translated to the interior. If the internal machinery fails to recognize and pass that message along, the brain remains trapped in a state of signaling instability. You can flood the exterior with SSRIs or sedatives, but the cell itself remains hyperactive. This translation gap is where a simple carbohydrate called myo-inositol operates. Historically categorized as vitamin B8, it is not a pharmaceutical drug, but a naturally occurring sugar alcohol that is highly concentrated in the human brain. Inositol functions as the mechanical bridge for this process. It provides the structural scaffolding required to carry those vital messages from the receptors on the outside to the machinery on the inside. Inside the central nervous system, myo-inositol is concentrated almost exclusively within the brain's support network, the star-shaped glial cells, or astrocytes. Within these cells, myo-inositol drives the PI cycle. When a neurotransmitter docks, it triggers a relay system. An enzyme splits the lipid into two second messengers, IP3 and DAC. Myo-inositol is the direct structural precursor allowing this split. Without adequate inositol fueling the PI cycle, neurotransmitters bind perfectly to the surface, but the internal machinery remains completely deaf to their signals. To see if optimizing this internal signaling treats severe anxiety, researchers tested massive therapeutic doses of inositol, between 12 and 18 grams daily. This chart compares the landmark Palatnik trial, pitting high-dose inositol directly against a common SSRI for panic disorder. Over 1 month, weekly panic attacks in the inositol group dropped by 4, matching or exceeding the prescription medication. Similar patterns emerged in the treatment of obsessive-compulsive disorder. High-dose inositol works as a highly effective standalone therapy for OCD symptoms, but interestingly, it provides no extra benefit when added on top of an existing SSRI prescription. This overlap reveals exactly how it operates. Inositol operates the same serotonin stabilization pathways from the inside that SSRIs target from the outside. For patients who cannot tolerate traditional pharmaceuticals, it provides an alternative route to the same destination. That intracellular stabilization also applies to sleep. Unlike traditional sedatives that force the brain into unconsciousness, inositol targets the metabolic fluctuations that trigger nocturnal awakenings. This graph visualizes 3 simultaneous mechanisms. Inositol boosts the production of relaxing GABA, maintains stable nocturnal blood sugar, and smooths out erratic spikes in cortisol via the HPA axis. But the strongest proof of inositol's neurological power comes from a completely different condition, bipolar disorder, and something known as the inositol depletion hypothesis. Traditional mood stabilizers like lithium achieve effects by doing the exact opposite. They actively deplete the brain's cellular concentration of inositol to halt manic episodes. The fact that lithium targets this specific cycle shows how strictly the concentration of a simple sugar dictates the extremes of the human mind, moving the dial from profound calm to clinical mania. As promising as the targeted trials are, the clinical reality requires precision. When researchers look at large-scale meta-analyses for general anxiety and broad depression, the results are decidedly mixed. To understand why, neuroscientists use magnetic resonance spectroscopy, shown here, to track exact metabolic levels in living patients. They are actively looking for who actually has an inositol deficiency. The data points to inositol as a highly targeted tool. It works best for a specific clinical phenotype characterized by signaling instability. Patients dealing with cognitive overactivation, severe stress intolerance, and persistent racing thoughts. When matched with the right patient, it achieves significant neurological changes with an incredibly mild side effect profile, avoiding the dependency risks and harsh impacts common to pharmaceutical treatments. The chemical environment between our cells is only half the story. The structural integrity of the machinery inside them determines if an overactive mind can finally hear the signal to calm down.